Okay, so good morning, everyone. Thanks a lot for coming. So we will start. So as usual, for those of you who attend on Zoom, you can report any technical issue on the chat and ask your question in the Q&A tab. And we will allow you to interact with the room directly during the discussion session. And with no further ado, I leave it to Mathieu to introduce the speaker. Yeah, you can go.
So good morning, everyone. So it's a great pleasure to welcome Tom Druet for this seminar. So just very briefly, Tom Druet is a senior researcher at the FNRS. So he works at the unit of animal genetics in the GIGA in the University of Liège. And his main research interests are quantitative and population genetics, mostly applied to domestic animals, and in particular, he has done a lot of methodological development regarding phasing or also studying the recombination landscape in a domestic animal. And what he's going to present today is his work that he has done on the inbreeding, on the characterization, the fine-scale characterization of inbreeding at the individual level. So, Tom.
Okay, thank you. So I would like first to thank you for giving me the opportunity to give this talk on inbreeding, understanding inbreeding with runs-of-homozygosity. So I first give a brief layout of the presentation, and I will start with some background on inbreeding and runs-of-homozygosity. And maybe some of you might already know these things, but I think they are useful to understand all the talk. I will briefly discuss about the methods used to detect runs-of-homozygosity. And then I have three applications, which are how we can use the distribution of HBD segments to understand demographic history, how we can use them to estimate the inbreeding coefficient, and then to study the effect of recent inbreeding versus ancient inbreeding. So I start with a very basic reminder, which is the definition of inbreeding. It results from the mating between two genetically related individuals. And so here on the slides, I have two examples of wild pedigrees from wild animals. And in both cases, you clearly see that the parents are related. And so inbreeding will be difficult to avoid. It will happen in all population, and it can happen at random. And so it will happen more often in populations with small effective size. And demographic events, such as, for instance, a bottleneck, can reduce this effective population size and increase the level of inbreeding. And other events like a breed creation can also reduce the effective population size. Then besides genetic drift, you have also non-random mating. Things like subdivision will increase inbreeding. Also selection, when you select individuals that have the same characteristics, they might also be more genetically similar. You have also some cultural aspects. When in a community, you only marry within the community. For instance, this is the example of the ancestry of a king in Spain. I guess the effective population size in Spain was huge, but they marry only among each other. And so basically, typically inbreeding will happen in domestic animals and in populations with small size. There are two main consequences of inbreeding. It's that increase homozygosity, which means more chance for homozygosity at rare alleles, and so genetic defects will be observed more frequently. And you have also a decreased heterozygosity, which means that you will have reduced heterozygosity for alleles that present an advantage at heterozygous state. So the general consequence is that you have inbreeding depression, which is the reduced fitness in inbred animals compared to other animals, and it's reduced survival or reproductive success. And it's important to know that there are two main hypotheses for this inbreeding depression. The first one is that the partial dominance inbreeding depression results from increased expression of recessive deleterious alleles. And the second hypothesis, which is less frequent, is the overdominance, where inbreeding depression would be due to the superiority of heterozygotes. In the first case, it means inbreeding will be due to rare and young alleles, and because you have purifying selection. And in the second case, you will have the alleles under balancing selection. They are maintained for multiple generations in the population at intermediate frequency. Inbreeding can be defined by, quantified by the inbreeding coefficient. It measures how closely related are the parents, and here is one of the two classical definitions, which is the probability that an individual carries two identical by descent copies. And so, so identical by descent means that, for instance, you inherit twice the star from the same ancestor to two different parts. And so because these two alleles come from the same ancestor, they are IBD. So what is interesting is that this allele is not inherited alone. In the original parent is on a chromosome, and so when you inherit the star, you also inherit an entire segment around the star. So it's completely homozygous, and therefore we use the term homozygosity by descent. So inbreeding will generate segments of homozygosity that will be around IBD alleles. And so these small segments that are homozygous, they can be observed as what we call like runs of homozygosity. Here is an example. I represent 5 individuals. Each line is an individual. Each number is a genotype, and heterozygous are coded with 1 and homozygous with 0 or 2. And so suddenly you have long stretches in the genome where you don't have any 1. So these are long runs of homozygosity, and this might result from inbreeding. So here is another representation that can help you to understand a bit. It's an example from a paper on mountain gorillas, where they represent only 1 chromosome. Each line is an individual. The different colors are the different populations. And so the small boxes are the runs of homozygosity they detected. And so with a representation like that, you can look at the frequency of the runs of homozygosity in the genome, but also the length, for instance. And the length is something important because the length of a run of homozygosity is directly related to the number of generations to the common ancestor. So it's related to the number of meiosis to the common ancestor. And Elizabeth Thompson wrote, for instance, in a review paper that the length of an IBD segment is distributed with an exponential distribution with a rate, which is a parameter from the exponential distributed, and it's equal to number of meiosis m. And so, which means that the expected length of an IBD segment is 1 divided by g, the number of generations. Now for an HBD segment, we have 2 times the number of generations to the ancestor because it's transmitted 2 times for 2 different paths. And so the expected length of a run of homozygosity or HBD segment, if we are in centimorgans, is 100 divided by 2g. So this is a nice property of a run of homozygosity or HBD segment. And so here I show you an example from a paper from Marty Cardos, where he looks at the distribution of runs of homozygosity in simulations for ancestors 2 generations back or 10 generations back. So what you see is that the average is longer for 2 generations, but you see also a lot of variation around the expectation. So the expected length is here, but you can have much longer or much shorter segments, and the short segments are in fact much more frequent. And a last slide for the introduction is, for instance, if you compare 3 populations with recent inbreeding, the ancestors are close, you will observe a lot of long segments. And if you have ancient inbreeding, you will see a lot of short segments. And so you will have a signature on the age of inbreeding. With this background, I can now speak a bit about how we will detect these homozygous by descent segments. So the most popular approach to do that is to use runs-of-homozygosity and PLINK. So it will simply count the number of homozygous markers. If it finds a stretch that is long enough with enough markers, enough length, it will declare it as a run of homozygosity. This is often used as a proxy for identity by descent, but there is no formal statistical support. So the idea is that if it's long enough, it should be IBD. And ideally, the parameters of this calling should be optimized for each data set. It will depend on the marker density you have, the area you use, the population you use, the allele frequency you have. A second approach that was already proposed a long time ago are likelihood-based runs of homozygosity. It still works by window, so we select a certain window size, and inside the window we compare the probability of the genotypes, if this is autozygous, by the probability to observe these genotypes if it's not autozygous. And to compute the probability, we will use the allele frequencies, also the genotyping error rates, Hardy-Weinberg equilibrium properties. And a last approach that is also quite natural is to use a hidden Markov model, where we will model the genome as a mosaic of HBD and non-HBD segments. So it means that the model will scan the genome and will try to identify run of homozygosity, and the start point can be anywhere and the end point also. So there are some properties in that model. It assumes that the length of segments are exponentially distributed. As the lot score approach, it uses allele frequencies, Hardy-Weinberg proportions, genotyping error rates. It's easy to extend the emission probabilities to use genotype probabilities or allele counts as we have with sequence data. So we don't need only genotypes. You can work also with low-fold sequencing or things like that. It used the genetic distance between the markers, which was not done by the other methods. And in fact, it doesn't use a given window size, but it integrates over all the possible windows, all the possible chains. And there is a probabilistic outcome. It tells you the probability to be HBD at each position. So in such a model, the age of the ancestors, as I told you, the length for the segments are exponentially distributed, which means the expected length is 1 divided by R, for instance. And as showed previously, the R should be a function of the number of generations to the common ancestor. So if you use one single R in your program, it will make the hypothesis that all the inbreeding comes from one ancestor or one group of ancestors present in the same generations. And in complex populations, this might be too restrictive. And so we extended this model to a model where you have multiple classes of HBD. So here is the idea. Instead of having only one HBD class in red, we now have one long HBD class and one short HBD class in orange. And every class will be defined by its own rate. So it means every class will have a different length. And it means that every class will correspond to a different group of ancestors and correspond to a different number of generations in the past. And so approximately, but really approximately, the age of the ancestors will be the rate multiplied by 0.5. And so also, this is not very important, but every class has its own frequency, a parameter that say often we see segments from each class. And the last point is that recently we modified slightly our model to improve its statistical properties, and now we model different layers of ancestors. We start with the first layer, for instance, ancestors present 5 generations ago, and in that layer we will use a model with 2 classes as the original model. But then the positions that are not HBD, they can be HBD from older ancestor in the next layer, and so on. So a few words of the benefits of the hidden Markov models. I just want to tell that we of course we compare this with other models, to this a bit the properties, etc. But I don't want to compare it too much here. Compared to the AirWatch with Blink, when you have a lot of information, both methods will give you approximately the same results. And the Markov models will be more interesting when you have degraded information. In case, for instance, when you have a lower marker density, a variable genotyping error rate, variable missing marker spacing, an example would be exome data, even variable recombination rate. And what is really nice is that you have a probability that indicates your confidence you are that this is truly an HBD segment or not, and you have less choice of parameters. So just to illustrate how it would work, if I take an example in dogs, here are 25 dogs, and I take the same representation as for the gorillas. So every line is one individual. We show HBD segments on chromosome 5, and we see that they have many more HBD segments than the gorillas, and an extremely long one, which is the full chromosome 3 here. So what the classification will do is to assign every of these HBD segments to a different class. So every class has a different color. And so here are the rates of the different classes. So it goes from 2 to 2000. The 2 are the very long segments, and 1000 are very short segments. And to help you, I put the expected length, which is more easy to read than the rate. And so, for instance, with a rate of 2, the expected length would be 50 centimorgans, and with a rate of 1000, it would be 100 kb. And so what we see here, for instance, is that we have the last individual as a very long segment is in the assigned to the class with the longest rate. And then you have 4 brown individuals, a lot of red and orange, which correspond to recent inbreeding. And then all small segments are painted in blue or green. Another way to represent that would be to look at the proportion of the genome that contributes in each HBD class. So for instance, we have a box plot where we have the values for the 25 individuals. And it tells you, for instance, that in the 3 classes here, there are the long segments, 10 to 50 centimorgans. It would correspond to ancestors present 1 to 5 generations in the past. There you would have all the short segments corresponding to very ancient ancestors. And what you see is that very recent inbreeding, you have most of the individuals have no inbreeding associated with recent ancestors, except 1. A few more for this class here, and then you see that the 2 classes in orange and yellow and maybe in green, they correspond more. They are present in all the individuals, and this might correspond to the breed creation of the dogs a few generations ago. So this is implemented in a R package Zoro, and now I will move to the first application is how we can look at the distribution of these segments to understand the demographic history. So this can be looked at individual level or population level. I start with the individual level. So if you look at the individual distribution of HBD segments, it tells you the information about the relationship between the parents, about the mating type in your population. As an example, in humans, based on the distribution of length, they will be able to tell you whether you have a marriage between first cousin or second cousin, which are traditional examples or more frequent example in human. And I go back to the example in dogs. So if we now represent the data as 1 bar per individual here, the total 8 of the bar would be the inbreeding coefficient of the individual, and you see that all the individuals have an inbreeding coefficient around 40%. But then you can add the color of the bar, telling you which classes contribute to the inbreeding. And what you see, interestingly, is, for instance, that individual 1 or 7, they have 25% inbreeding associated with a very recent ancestor, very long segments. And this would match what you expect for a parent offspring or a full SIP matting, because when from pedigree data, the expectation would be 25% inbreeding, and with parent offspring, your rate should be 3, and with full SIPs, your rate should be 4. So this might indicate that in this data, there was maybe a mating between 2 full SIPs or a parent offspring. And as a comparison, you can look this individual here. He has the same level of inbreeding, but there is no long segment, no red, no brown, no dark brown, and so it's the ancestors are older for that individual. And so just as a comparison, I made the same in using grid data from the UK, and there are many more individuals. What you see here is that there is almost no inbreeding in that population. So, and in particular, no recent inbreeding. You can have background inbreeding, but even like that, it remains very low. But then you have a few exceptions, like I would say accidents, where you can detect one individual, for instance, with 20% inbreeding with reddish colors. So it helps you to understand what's going on in the population. Now I move to the population level, and I start with a figure from a review by Ceballos et al. And it shows you, it compares the number of runs of homozygosity and the total length of the runs of homozygosity, and it says that this will reflect the history. So this is I would say the large standard population. You would be here, and if the population gets smaller, you get more runs of homozygosity. If you get a bottleneck, you get even more runs of homozygosity. And if you have admixture, the number of runs of homozygosity will go down. And if you have consanguineous mating in your population, you will tend to have a few longer runs of homozygosity. So what is very popular for a few years already is then to represent a classification of the runs of homozygosity in different length classes. This is an example in human, where you have populations from the different continents, and the most interesting populations here are probably in red. Oceania is the population which has the most short segments from 0.5 to 1 megabase, and then it goes below the orange, and it goes even below the green ones. So that population has the most ancient inbreeding, but not the most recent, and it's related to a founder event in Oceania when the population arrived there. Now the orange population, they have a lot of recent inbreeding too. These are Indian communities in South or Central America where they have some consanguineous matings. So here is another example in Indian tiger. What I want to show you here is more that it starts to be common to translate directly the length of the runs of homozygosity in a certain number of generations. And I think this is a bit dangerous because we saw that there was a lot of variation around, but directly they even compute the number of generations in each class and transform this to an effective population size. And so a question was, how good is this kind of approach? And there we used, we analyzed data with our package ZOO or with ROH-based classifications on simulated and real data. And for the ROH, we selected classes that match the HBD classes from Zoro. So here is the first simulation, which is a very simple simulation where we model the genome as a mosaic of HBD and non-HBD segments. And the length of the HBD segments, the expected length is 12.5 centimorgans. And we try it with 4 values of inbreeding from 0.05 to 0.4. So what we see with the ROH classification, the theory or the expected length would be inside that class. but we observe that other classes also contribute to the inbreeding, and that there is a shift to the left, and the shift is more pronounced when the inbreeding levels increase. With the hidden Markov model in this simple situation, it always identifies correctly the class that contributed to the inbreeding. Then we move to a bit more realistic simulations, but still quite extreme, where we modeled a very strong bottleneck 64 generations ago. And it's with a simulation software under White-Fisher process. And what we see here is that with the hidden Markov model, again, you can identify correctly the class that contributed to the bottleneck. And with the ROH classification, again, you have a shift to the left, and the correct class is not clearly identified. Of course, this is quite approximative, and we show also examples using the hidden Markov model with more complex histories where we would have two distant or close bottlenecks. In that case, we still can guess a bit the history, but it becomes less precise. For instance, with two close bottlenecks, it will concentrate the inbreeding in the middle of it. And if with the bottleneck followed by an expansion, you see when the bottleneck occurred, but you cannot clearly see the history. So now I will illustrate this with 4 examples that are on the slide. I start with the European bison, and you should know that it has been extinct in the wild around 1930. And it has been restored after that from 12 founders only, which were animals from zoo and breeding centers. And after that, they've been reintroduced in the wild, and there are more than 2,000 individuals in the wild now. And this is really an example of severe bottleneck. For the cattle populations, we have 3 examples of populations. A Dutch Holstein cattle, which is a typical cattle population that has been selected recently, quite intensively. Zebu cattle from Madagascar. And then a quite nice example from feral cattle from Amsterdam Island. It's a small island here south of the Indian Ocean. And in 1871, there were 5 or 6 individuals that were introduced on the island. And they were abandoned there. And the size of the population increased quite rapidly to 2,000 individuals. So when we look at these 4 populations and we estimate the effective population size with a program called GON based on the leakage disequilibrium, we see what we expect, that in the last 10 to 20 generations, the Holstein cattle, the effective population size reduced to approximately 100 individuals. For the feral cattle, we see that we have a very strong bottleneck below 10 individuals 15 to 20 generations ago, but that after the bottleneck, the size of the population increased again. For the zebu of Madagascar, we have a much less pronounced bottleneck, but over many more generations and in older generations. And finally, for the bison, we see the bottleneck, but it has not completely recovered. Now, if we look at the distribution of HBD segments in all of these populations, we see that they reflect quite nicely what we observe in Holstein cattle. We see that in the 15 last generations, a little bit of inbreeding has been accumulated, but not so much, because the effective population size is still around 100. Here we have a really strong signature with HBD segments tracing back to the generations that match exactly to the bottleneck. And so we see that all the inbreeding, and it's quite massive, 30% inbreeding tracing back to these generations, but there is no recent inbreeding because now the population was larger. For the zebu of Madagascar, we see that during this long bottleneck, a lot of inbreeding has been generated, but it was very small segments. And finally, for the bison, the inbreeding is more recent than for this one, but the levels are higher, and there's a bit more inbreeding in recent generations. If you do the same with ROH-based classification, for these 2 examples here, we see that, again, the picture is less clear with a shift to the left. And the last application based on the distribution of the segments is that you can look at the dynamics or the evolution of the inbreeding with this approach. Here, for instance, we can compare the inbreeding in individuals that are born before 95 and after 2005, the level of inbreedings are approximately the same, 0.40, but the distribution is a bit different because individuals born after 2005, they have more short segments. So it means that it now takes more generations to trace back to the common ancestor, and that there is less new inbreeding events generated in the population, which is quite positive. I can show this also with graphs where we look at the trend as a function of the year of birth. We see that inbreeding is stabilized, but now we have more and more short segments and fewer long segments. So it means that the population is going into the right direction and that we can use this distribution to monitor the evolution. Now I will move to the second part of the application is to show how we can use HBD segments to estimate the inbreeding coefficient. So before showing how we can do that, I would just say that there are many different estimators of the inbreeding coefficient. The first one is the pedigree-based, which is based on the genealogy. Then you have those based on the HBD and ROH that I will explain briefly. Then you have a lot of molecular approaches, which are all a bit different, based on the united gametes, on the homozygosity, on the genomic relationship matrix, on a maximum likelihood estimator. And so they have all different properties. And we give a few explanations in a paper here. So with runs of homozygosity, the inbreeding coefficient is simply computed as the sum of the length of all the segments divided by the sum of the size of the genome. And with the hidden Markov models, you can do similar things. A very nice feature with that is that the inbreeding coefficient ranges from 0 to 1 like the pedigree one. And the values are also interpretable, just to tell you that united gametes or JRM or excess homozygosity, they can go from -1 to 1 or even beyond 1 or -1. And it's not always easy to interpret. With a multiple HBD class model, you can even know the proportion of the genome in each class. And so I illustrate this in Belgian blue cattle, where we have 600 sire genotype for 600,000 markers. I show you first the effective population size to understand the data. So it has been declining in the last 10 to 15, 20 generations ago. And here is a zoom just to say that in the last generations, it's around 100. Now with that, we fit a model with 13 classes that are indicated above. And so as before, we have the recent inbreeding here and the very ancient inbreeding there. And after the bottleneck, we can say that some inbreeding has been generated in the classes 8 to 32. And also that we have no recent inbreeding in the classes 2 or 4 with some outliers. So when you use the runs of homozygosity or HBD segments to estimate the inbreeding coefficient, you can use the length of the segments because you know it's related to the number of generations. So you can use the length to select your base population when you estimate the inbreeding coefficient. So for instance, here, if I take back this picture where we have all the segments from all colors, we could decide to say that we want only to have an inbreeding coefficient for the last 10 generations and select only segments that are from the first 4 classes like that here. If you include everything, you would go back many generations in the past and it would maybe not be too meaningful. So here is a representation of this. We could start with only 4 classes here. This would correspond to a certain pedigree depth. And you say, okay, now I want to add one more class, the class 42. And so I had all the yellow segments now. It would correspond to a deeper pedigree. You could add also the green segments here, and then your pedigree depth is even higher. And finally, you can add all the segments, and then it would be the entire pedigree. And so if you do that in Belgian blue cattle, for instance, you have no inbreeding if you include only the 2 first classes, which is normal. And then if you include 10 generations, you would have on average 5% inbreeding and around 10% 100 generations ago. And so we could ask, so I am presenting you the use of runs of homozygosity as a estimator of the inbreeding coefficient. And it's interesting to know how they compare to the other inbreeding coefficients. And in the literature, there are some contrasting results. There are papers telling you that united gametes is the best, and others that FOH is the best. So we made an evaluation with real data and evaluations based on simulations. So in the real data, we have whole genome sequence for 260 bulls. And the idea was to say, okay, if you have the whole genome sequence, then you have all the information, you know, all the homozygosity of the individual at all the kind of alleles, etc. And so we will estimate the inbreeding coefficient with only 40,000 SNPs and compare to what we have in the sequence. And so there we have four graphs, and each color represents one of the methods we compare. And the first graph here on the top left looks at the relationship between the inbreeding coefficient and here you have all the allele frequency, and we look homozygosity. So if we are interested in alleles causing inbreeding depression, I said this should be rare. So we should look most on the left here and check which is the method that captures best homozygosity at rare alleles. And there you see that united gametes, or JRM, the blue ones, are among the best, and that HBD segments do a bit better than ROH or homozygosity. But as I told you, the deleterious alleles should also be young. And so to enrich our data set in young alleles, we selected only private alleles, so that are not observed in other breeds. So because alleles observed in other breeds are old, so we take only those that are not observed in other breeds. And this is the second figure. And we see that in that case, for alleles around 10%, the methods get much closer, so that for rare and young alleles, difference is less strong. The third graph looks at heterozygosity. So SNPs presenting heterozygous advantage should be here. In that case, the blue and the red, they are quite good. And also we see that these alleles are more easy to capture than the homozygosity at rare alleles. And finally, the last graph is the correlation with the whole genome heterozygosity or homozygosity. So it tells you which method correlates more with the total diversity at all alleles of the individual. And in that case, ROH, homozygosity, and HBD are much better than united gametes, for instance. So in the same study, we also looked which is the best method to capture homozygous mutation load. So this is the homozygosity at rare alleles. So it's to mimic recessive deleterious alleles. And so we compared for alternative and private alleles again for different, here it's always the same allele frequency, but different functional classes. Synonymous variants in gray, tolerated variants, missense variants in orange, and deleterious variants in blue. So first, I think we should look on these alleles. We see that the difference between the methods, for instance, HBD, get as good as united gametes for the deleterious alleles, and they are quite close even for the synonymous variants. And for alternate alleles, not only the young ones, then the united gametes is a bit superior. What we see also is that the correlations for the SNPs that best match deleterious alleles are not so good. So in parallel, we participated to a study in done by Armando Caballero, and just to focus on the most interesting results is that he looked at the bias in the estimation of inbreeding depression with the different approach, and the most interesting are the best ones, and the most interesting are the green one, and the last ones, which are the runs of homozygosity. And so he started by doing simulations above with an effective population size of 100, and he ended up with effective population size of 10,000. And with 100, ROH-based approach are unbiased, and the united gametes is a bit biased. And when you increase, the united gametes is not longer biased, but the ROH start to be biased. So the conclusion is that the best approach might depend on the effective population size you are studying. And he also shows that the correlation with the fitness is best with united gametes. So to sum up this comparison, it's which is the better approach might depend a bit. We saw that united gametes is the best at capturing rare alleles, but we have smaller difference when you focus on rare and young alleles that should look like the deleterious one. It depends on effective population size, and with effective population size, when you have smaller effective population size, deleterious alleles can reach higher frequency also. So then the methods get more closer, and hitchhiking can also bring deleterious alleles to high frequency. And it depends also a bit on your objective, if you want to limit inbreeding at deleterious alleles or at all the alleles. So this is just an empirical test of this in Belgian blue cattle, where we had 14,000 genotype codes with 28,000 markers. And we estimate the inbreeding depression. So it's a regression on the phenotype on the inbreeding coefficient, and we look with different methods. And just in that comparison, the HBD segments was kind of performing the best, the most associated. This is the strength of association here on the right. So it's the best p-value. This is the inbreeding effect. So it means that the completely inbred individuals would be 20 centimeters shorter. But so this indicates that indeed, with empirical data, in a population with small effective population size, HBD is a good estimator, but the united gametes was quite good also. Another nice property when you use ROH to estimate the inbreeding coefficient is that they are more robust to allele frequencies. So most of the methods, molecular methods I presented, they need to have the allele frequencies, and you need to know the allele frequency in the base population. But it's difficult to have the allele frequency in the base population, in particular in white populations. You need a pedigree, and you need genotypes from ancient individuals. And so just to show that this might have some impact, here we compare genetic trends for inbreeding from year 2010 to 2020 with 4 different estimators. The green one in the middle is the pedigree, so it doesn't use allele frequencies. The red one, we see no difference. It's based on the HBD segments. But the 2 other methods here, molecular methods, united gametes and GIGA-R, we see that they give different trends if you didn't use different allele frequencies. Here it's with the sample allele frequencies, and here is with the base population allele frequencies. So when we use base allele population frequencies, we have the same trend with everyone, but we see that these 2 methods, they will give you different results if you don't use the right allele frequencies. And so here are correlations when you use the population allele frequencies or the base population allele frequencies. And there are many studies comparing all the inbreeding coefficients using sample allele frequencies, and then the conclusions are that some are really not well correlated with the pedigree, they are not highly correlated with each other, but when you use the base population allele frequencies, the correlations, they get much darker and they get much higher. And so the last application I want to discuss with these methods is that we can use this inbreeding coefficient to compare the effect of ancient versus recent inbreeding. So this is the last application. And so the hypothesis is that purging should remove deleterious variants from the population. And so ancient inbreeding should be less deleterious than recent inbreeding. There have been some studies in livestock populations with some evidence from pedigree data and less strong evidence from ROH. And a recent study also in wild populations, the soy population from the group of Pemberton. So they classified the runs-of-homozygosity in long, so the recent ones, the medium, and the short ones. And so this is the distribution in the population. So for instance, the short ones, they contribute 10% of the genome with little variation. The long ones, they are quite rare, but some individuals have a lot. And then you have some 20% due to medium ones. And when you look at the effects on first-year survival, what you see is that there is no effect of the ancient inbreeding, medium effect of the medium inbreeding, and a negative effect, reduced survival for long inbreeding. So we tried to do the same with our cattle data. And so we used 10 classes. And I think it's important to look at the distribution in each class, how much variation we have. So we have the two first classes where there is little variation. And for instance, this class has not as much variation as this one. And when we do that, we have a quite nice figure. So we fit the inbreeding coefficient of, the effect of each class simultaneously. And the effect is more negative for the recent inbreeding, which is on the bottom. And there is no effect for the ancient inbreeding, which is on the top here above. So this is the effect size, and the significance is here. So the four classes here, which represent ancient inbreeding, are significant, and these ones are not. But we wanted to go a step further for this, and we thought maybe the results we get are just an artifact because we don't have enough variation to estimate inbreeding in ancient classes. And also the ancient inbreeding is estimated with less precision. So we start by doing a simulation where we are going to simulate constant inbreeding for all the classes. And with our data, check what result would we get. So for instance, if we simulate constant inbreeding, we get the gray box plot, and we would get an effect around -22 always with all the classes. And in all situations, for the ancient inbreeding, we would get higher effect than what we got in our higher effect that we got in our simulation. So it might indicate that if the inbreeding effect was constant, we should observe higher effect. And then the second graph shows you the power we have to capture the inbreeding. And so what we see is that in all 100% of situations for the intermediate classes here, we always detect the inbreeding. For the ancient classes, we have a bit less power, but still, in most of the cases, we detect something. We made a second thing to try to improve our analysis, is that we thought, okay, that in these classes, 128, 256, I think we would get like 4 or 8 SNPs per segment, and we might not estimate this very precisely. And so we wanted to bring more markers, and this was done by imputing to the bovine HD, which is 20 times more markers, and we obtain the same results, which is quite good. With this, I will move to my conclusions, and I would start with a comment. I didn't review all the applications you can do with runs-of-homozygosity. What is done more and quite often now is to use runs-of-homozygosity to make a GWAS based on runs-of-homozygosity to try to find regions that cause inbreeding depression or to find deleterious alleles. But I think people should be careful of that because that method will be superior to other approach only in very specific cases, which would be, for instance, to have multiple deleterious alleles in the same region, or that all the homozygosity is deleterious. If you have just one single deleterious alleles, it's better to do a scan for recessive effects for dominance effects. The second thing that is done a lot, at least in my community of animal breeding, is to use runs-of-homozygosity to detect signatures of selection. And it's true that runs-of-homozygosity, it's related more or less to extended haplotype homozygosity, for instance. But I'm not aware of any methodological validation that shows that this is a good approach. And I'm not aware of a paper showing that this approach is superior to existing approaches to identify signatures of selection. And if you don't have these two things, you should not use it. So to conclude, I try to show you that the runs-of-homozygosity are informative about the demographic history, but this is only an approximation, and in particular with length-based classification of runs-of-homozygosity, we should be very careful. They can also be used to understand the mating structure or look at the dynamics of inbreeding. And the second part is that the runs-of-homozygosity or HBD segments can be used to construct a very reliable estimator of the inbreeding coefficient that is interpretable, and it allows you also to study the age effect of inbreeding, and also to identify regions contributing to inbreeding depression. So with that, I will end and thank all collaborators on this for all most of the methodological part. We work with Mathieu here. Then there are all the people from my group, and in particular Molona and Natalia that helped me with this work. We had nice collaborations with Armando Caballero and Laurence Flory, and with people from Poland for the bison. Thank you.
So thank you very much, Tom. So I don't know if somebody has questions. Plenty of questions.
Hi, thanks for your talk. What are the limitations of your runs of heterozygosity analysis? Like how low can you go? Can you go below 100 kb? And how far back can you go in time, like in terms of generations?
Thank you. That's a good question. So how far you can go depends on the marker density you have. So with runs of homozygosity, in Pling, for instance, theoretically, you should have regions of at least 50 SNPs. So you can make the count. So say 50 SNPs, how much would we represent with different genotyping or sequence data? And so with the Denmark models, you can also go, you can even capture inbreeding with a single SNP based on the allele frequencies. But so the efficiency or the accuracy will go down, and I would say that you should not overinterpret too much segments when you have less than 10 SNPs per segment. And also then I think these are tools more, they should not be used to study the population ancient history, because then you have tools that are specialized for that, and they're really focused on the inbreeding, and I would use them mostly for maximum 100 generations. I think that it's for the recent generations that they are the most meaningful.
Yeah, thank you. So maybe a question I had is, so I guess that these the length distribution of these runs of homozygosity is based on a random recombination in each generation. So what would happen if recombination is not random in species that have hotspots of recombination, like this with PRDM9? I guess the decrease of these slower.
Well,
so maybe you have some ideas about that.
Well, so I would say that PRDM9 would mainly affect local variation in recombination rate. And so if you look really at very fine structure, fine locations, very extremely small segments, maybe it will have a lot of, it might have impact. But when you start to look at segments of 100 kb or 1 megabase, then the effects of the hotspots are average in the window, and also the method takes into account the recombination rate. So I think what would be a problem with regard to your question would be more that the recombination rate change to the generations, but I'm not aware of something doing that. But the variation along the genome is accounted for, at least for relatively long segments. So if you go very small, maybe that might have an impact, but if you are in large windows, not.
Well, just related to that point, actually with PRDM9, there is covers of PRDM9 alleles,
yes.
And so when you exhaust hotspots, then
yes.
Then of course when you get a new allele, then it completely reconfigures the recombination landscape, and so that might be a case where
might create maybe variation.
Time.
I don't know how much variation in recombination rate it would create, because you always need enough recombination, because if you don't have meiosis or you have too much recombination, you have problems. But it might create a bit fluctuation and make this estimation of the age a bit less precise. Yes.
But my question was more about the how do you deal with structural variants that might occur in genomes that might be polymorphic?
Okay, well, structural variants can have different impacts. If you have an inversion, it can impact the recombination rate, or they can also make your data more dirty. I mean, the coding of the genotypes, etc. So I would recommend for this approach is to clean your data as much as possible. When you have sequence data, you don't need to have all the markers. You have so much information that I would filter the data to avoid having the impact of CNV. I would ignore them and remove them. And so I think their impact on recombination should be quite local and not too big, except for large inversions. But even the large inversions might be accounted for if you include them in your map. I don't know.
So no question on the chat, so I can ask a question. When you say that ancient inbreeding contribute less to inbreeding depression, is it just because runs are smaller, so they contain less deleterious mutation, or does it mean that you selection against deleterious alleles in the chromosome tract have been removed generation after generation?
It would rather be the second hypothesis. Well, the second part you say would be the main hypothesis for that, because the length of the tract, it's a regression. The regression coefficient, the value it tells you for the same amount of inbreeding. So this is accounted for, that there is more or less proportion of the genome in the different tracts. But I think that we have to be careful, and this must be validated, because what might happen is that if in the ancient classes you have less information, less variation among individuals, you make more errors. You might have less precise estimation and conclude that there is no longer effect, but in reality there is, and you are not able to detect it. But the basic hypothesis is that the short segments means that you trace back many generations to the ancestor, and that the old alleles they have been removed from the population, and deleterious alleles, because they are young, they can only be on the long HBD segments.
So they are both shorter and they carry less deleterious mutation in.
Yes.
Okay.
The strength of those approaches based on HBD segments and RRH is that they don't rely on the knowledge of phase information in the genome, so that they can be accessed directly. But now that this information about phasing becomes available in genomic data sets, how do you think that the knowledge of genetic ancestry across recent pedigrees can help to study the impact of inbreeding, for instance, by providing direct access to the age of ancestors, avoiding confounding effects of recombination, variation along the genome?
So I'm not sure to fully understand the question, but one part is that, well, with inbreeding, you don't need to phase. Yeah. But you think that with accurate phasing, you can get better information about the ancestry?
Yeah, that analyzing phased genome would help to reconstruct the ancestral recombination graph, and that HBD segments are basically summary statistics of this graph. So if you can access directly to that structure, then you can identify more directly the age of shared ancestor from which yes. HBD segments are inherited.
So I would say that that when the methods to detect IBD segments get more reliable, you have, of course, much more information in IBD than HBD, yeah. Because if you have 10 individuals, you have only 10 information from 10 HBD segments, and when you have IBD, you have all the comparisons, and there you would have more information, and you would have more information to infer the past demographic history, etc. And you can also have more information to study how frequent are deleterious variants on long IBD and short IBD tracts. I agree with you in that sense that HBD is, how should I say, it's practical because it's concentrated, and maybe you lose some information when you want to study a population.
So we have a question from Simon Boitard. You can, we can hear you now.
Hello, can you hear me?
Yes.
Yes.
Yes. Okay. Thanks to take my question. Thanks, Tom, for the nice talk. I have a question a bit related to the previous one. I was wondering, as at some point you showed a comparison between the estimations of recent NE by the method GONE and the HBD distribution that you estimated. I was wondering if you pushed a bit further and tried to reconstruct such histories of recent population size using summary statistics from your HBD distribution.
Yes, we started to try to do that. So because once you have the proportion of inbreeding created in one class, if you know exactly the number of generations that are captured by the class, you could ideally, you could in theory estimate the effective population size. So the difficulty is to know exactly which classes, which generations are captured by which classes. And well, I think that this approach will be probably less precise than other approaches, but might be complementary. Also, it will depend on how you define your class's boundaries, because if your class exactly match one bottleneck, then you might estimate the effective population size in the bottleneck. But if the class you define is twice the size of the bottleneck, the inbreeding will be diluted in more generations. But yes, we are trying to work on that to know which generations are captured by different classes and how it translates to any. And maybe on that one I might need help, and I might ask you at one time. No?
Yeah, I think one may not necessarily need to have a very strict correspondence of HBD classes to a given time, but that could be something more a bit more kind of a black box, like using the HBD distribution as summary statistics and just see how these summary statistics globally contribute to estimate a past any profile.
Yeah, you could use the summary statistic in an ABC approach, you mean?
Yes, yes.
Yeah,
I was
that's
thinking
also a possibility, yeah.
I was thinking of potential applications of the method for plants, where there's decline in pollinators or stuff like that, where you might expect recent changes in mating regimes, like increasing selfing or stuff like that. And I was wondering whether you could use that method to estimate the rate of change in mating system in recent history, or estimate the variation of selfing rates through time, something like that. Do you think you can do that?
It's a difficult question, and a very good but difficult one. Well, I don't know what exactly means the rate, but I think you can. I think there should be opportunity to look at the different distribution in different populations to see whether there are changes, and that the different classes can help you. About selfing, we already discussed it several times. Maybe it could be used to estimate the rate of selfing, but there are other approaches, I guess, that already do that. And I think if there's too much selfing or too much inbreeding at one moment, it starts to be very hard, because if an individual is completely inbred, it's difficult to know how many segments there are. So so if it's too high, it would be difficult, I think.
I guess the change in selfing rates that people are concerned about is with plants that have a mixed mating system. So generally, the mate is not like pure selfers. It would be like species that are outcrossing mostly, but might have seen an increase in selfing due to the lack of pollinators or other change in the population.
An event of selfing should generate very long segments, and maybe it might be possible to classify self-done plants and non-self plants, and then to see whether this frequency increase. But, it's difficult to give a very good answer. I think it needs more development, but yeah.
So, and we have a last question online by Patrice David. Patrice?
Yes, can you hear me?
Yes.
Yes, I had a question regarding the use of the method for like bottleneck populations or populations that have been recently to very small sizes. So there will probably be in the genome some places where there is segments that are completely fixed in the populations. And so when, does this count as a run of homozygosity if it's shared by all the individuals in the population? Or if the runs of homozygosity are detected based on differences from random mating, then they would not be detected by, in that case, if they are shared by the whole population?
Yeah, that's a good question. So what will happen is if you compare with runs of homozygosity like those in Plink, so if all the population is fixed, but you keep the marker, it will be considered as a run of homozygosity. And if you clean the markers and you remove the rare alleles, it will be not detected. And so with Plink, it's important to know whether you can remove the fixed alleles or not, because sometimes I think we should remove alleles that never existed in the population. So with the HBD approach, it will depend on the allele frequencies you use. If you are able to have the allele frequencies in that region before the bottleneck, then you can detect it and say this is HBD and this is ancient HBD at the time of the bottleneck.
So then you require a reference population that you know from another sample.
Yes, for instance. And if you don't know the allele frequencies and you use the allele frequencies from the sample, these regions will not be informative and it will be declared as run of homozygosity or HBD only based on what's happening on the neighbor regions and not in the region, because if it's fixed, we don't have information. It means like we don't have SNPs to estimate it. So we need to detect a sweep with that approach. We would need to have the allele frequencies before the sweep.
Okay.